Hello, my name is Crystal Vander Zanden, and I am an assistant professor in the Department of Chemistry and Biochemistry at the University of Colorado, Colorado Springs. Today I'm going to talk about some of the research I've done in my postdoc and some of the research I've done during my first year as a faculty member. The title of my talk is X-ray reflectivity and grazing incidence diffraction studies of amyloidogenic and peripheral membrane binding proteins. I'm going to be talking about these X-ray scattering methods applied to two different biological systems. The first are amyloidogenic peptides involved in Alzheimer's disease, and the second is the peripheral membrane protein 15-LOX2 that binds membranes and oxidizes lipids. So first, why is it interesting to study the structural biology of cell membranes? I grew up doing my PhD in crystallography, doing crystallography of proteins and nucleic acids. And the membrane, by comparison, is a much more complex system with many different types of lipids, with different other molecules like sterols and these membrane proteins. It's a very complex, very dynamic system that makes it really interesting to study. The structure determination methods for membranes are not as straightforward. Though we do have some things available to us like lipidic phase, crystallography, or electron microscopy, but it's really not as straightforward as simple X-ray crystallography that can be done for proteins or nucleic acids. And even for proteins, you need a very well-behaved system to achieve crystallography. So it's really a misnomer to call that simple. But the membrane is in its own nature a fluid system, so it's difficult to use these static structure techniques such as X-ray crystallography. And the biophysical characterization is also really important. So you'll notice in this diagram, this lipid membrane is split into two distinct phases. The liquid expanded phase, or LE phase, where each one of these lipids is taking up a little bit more space. The tails are often bent instead of rigid, and it's much more fluid. The lipids are moving around. And that's in contrast to the liquid condensed phase. And you'll notice these lipid tails are straighter, the lipids are packed in closer to each other, and these phase dynamics often help sort proteins into the correct part of the membrane. So some proteins prefer to interact with, for example, the liquid expanded phase or the liquid condensed phase, or perhaps at the boundary of the two phases. And this phase separation is very important for how the cell modulates its activity. Everything starts at the cell membrane, so this basic biophysical characterization is very important. 70% of pharmaceutical drugs target membrane proteins. The membrane acts as a semi-permeable barrier to only allow selective molecules through. So what this means is that if we're trying to target something in the cell, the easiest way to do it is to target a protein on the membrane because many drugs will not pass through the membrane. So this just shows the percentage of different drugs on the market that target different regions in the cell, where intracellular is only this portion here, and the rest are things at the membrane or secreted outside the membrane. So overall, the membrane is really important for druggability, but also membrane dysfunction is involved in a number of diseases, particularly in this function of it being a semi-permeable barrier that plays a big role in Alzheimer's disease, which I'm going to discuss today, as well as other neurodegenerative diseases that have a similar mechanism. There are others, of course, and one that's on our mind all the time these days is for the SARS-CoV-2 virus to gain entry through a receptor on the membrane. So many facets of cellular behavior are regulated at the membrane. The final point is that lipid diversity is sort of an unsolved puzzle compared to other building blocks of life. Like when you teach an introductory biology class, we teach that there are 4 building blocks of life. There are 5 nucleic acids. We know them, we know their structure, we know their function. It's fairly well understood. 20 common amino acids. There are other uncommon amino acids, of course, but Life gets most things done using these 20 common amino acids. Lipids, on the other hand, there are hundreds of different types of lipids, and they vary organism to organism, tissue to tissue, and within different cellular compartments, the plasma membrane versus the mitochondrial membrane versus the ER, for example, and they even differ between the inner and outer leaflet of the same membrane. Lipid complexity is very interesting. We have all these different head groups and different tails, and they must be serving some function in biology, and generally we don't have a good grasp on these different types of lipids. So now the biophysical methods that I use to study membranes. One of our workhorse methods is the Langmuir Trough, and the Langmuir Trough is a dish where you can deposit lipids with a large area per molecule. And in this initial state, the lipids are considered to be in a gas phase, not really interacting much with each other. And then as we compress the lipids, we decrease the surface area per molecule, and we see an increase in surface pressure. So by this, we can mimic the pressures that would be found in a native cell membrane. And this allows us to do very careful measurements, determining how lipid behavior responds to different temperatures, pressures, or lipid compositions. We can also use a Langmuir Trough to determine protein interactions with the membrane. So we have this Wilhelmy plate submerged into the surface that forces are measured on it. So this can detect increases in surface pressure. So one way to do these experiments is you have these barriers fixed at a constant area, and then you inject proteins into the subphase. And as you have more material gathering at the air-water interface, like proteins binding to your monolayer, this is going to cause an increase in surface pressure over time. You can also do these experiments using constant pressure and then allowing these movable barriers to change the area in response to protein insertion, but all the data I'll show today was performed under constant area conditions. Another method we use is fluorescence microscopy, where we have our membrane and we dope it with about 0.5% of a Texas red labeled lipid. And Texas red is this bulky fluorophore, which prefers to interact with the lipid tails, the hydrophobic lipid tails. And because of that, it partitions into the liquid expanded phase, where there's more room for the fluorophore among the lipid tails. So what that means is that these dark domains consist of liquid condensed phase surrounded by a brighter liquid expanded phase surrounding it. So we can use fluorescence microscopy to see how these domain separations change based on protein interaction. So we can look for things like the shapes of these domains. Are they fusing, which may indicate decrease in line tension? Are they becoming rougher around the edges? These are pretty smooth, well-defined. What's in the condensed phase, what's in the expanded phase, but sometimes we'll see those boundaries become less distinct. Or is the protein changing the overall amount of LC versus LE phase? We can probe those questions using fluorescence microscopy. The next method is X-ray reflectivity, where we have our monolayer set up on a Langmuir trough, and X-rays are reflected from the surface. Our detector here detects the intensity of X-rays that are reflected, and we plot those normalized to the ideal Fresnel reflectivity. And that Fresnel reflectivity is a function of the electron density as a function of the depth through the monolayer, Z. But we can't explicitly solve for electron density, so instead what we do is we build models constructed of different slabs, where we have a slab with one particular length, electron density, and a roughness parameter in between the slabs. So we build these separate slabs to develop a model, and then we iteratively refine our model until we get the best match with our data. So by doing this, we can get an idea of what the membrane looks like on its own, and also how the membrane changes in response to the addition of protein. For example, if protein were to bind, interacting with the head groups here, we would see the addition of electron density beneath the head groups. The last technique is grazing incidence X-ray diffraction, where we're still working with our monolayer on a Langmuir Trough, and this time we're diffracting X-rays from semi-crystalline species present at the air-water interface. These semi-crystalline species include, for example, lipids in the liquid condensed domain. And from that diffraction, we can fit that integrated curve with, typically we use a Voigt profile, which is a convolution of a Gaussian and a Lorentzian. And from that fit, we determine the position of the peak, which can be used to calculate the lattice spacing between items in our semi-crystalline species. The full width half max of the peak is used to calculate the coherence length, or the average length of coherently scattering objects. And the area of the peak is proportional to the amount of semi-crystalline material. So for example, if we add protein and we see that peak area decrease, we know that the protein is relatively decreasing the amount of semi-crystalline material. So as I said, I'm going to be talking about those methods and applying them to two different biological questions. We'll start with the question of these amyloidogenic peptides that are involved in Alzheimer's disease. So what do I mean by amyloidogenic? Amyloidogenic are peptides or proteins that have some intrinsically disordered components and no stable persisting structure. And because of this instability, it makes them really prone to aggregate into more stable structures that can be toxic to the cell. So one of those stable structures are amyloid fibrils, which are really rich in beta sheets. So that's what we mean by amyloidogenic peptides. I want to start by thanking the other folks who have been involved in this project. This is work I did during my postdoc with Dr. Eva Chi. She was a wonderful postdoctoral mentor. Dr. Yaroslav Majewski was also involved in a lot of the data collection, analysis, and discussion of the results. And also two undergraduates I trained in the summer of 2018, Lois Wampler and Bella Bowers, were also involved in this project. And of course, this was done at NSF's ChemMatCARS Sector 15, and my postdoc was funded through an NIH ARACTA fellowship. I won't belabor this point, but I just want to briefly say that Alzheimer's disease really is one of the greatest health issues of our time in the US. Approximately 1 in 10 people will develop Alzheimer's disease in their lifetime. And of the top 10 leading causes of death in the US, it's the only one that we have no way to treat, no way to prevent, no way to slow it down. So we really do need a lot more information on the molecular mechanisms of this disease. Neurodegeneration in Alzheimer's disease, where neurodegeneration means the death of neurons. And we see this on a macro scale when you image a person's brain post-mortem after they've passed with Alzheimer's disease. You find these large cavities in the brain from these neurons dying, from these tissues dying. You can weigh the brains and you find that they physically weigh less in mass. Alzheimer's disease has two classical hallmarks. One is the presence of A-beta plaques, or these protein-rich deposits present when we do histology on brain samples, and also these other protein deposits called neurofibrillary tangles made out of tau. So A-beta plaques are extracellular, and tau tangles are inside the cell, sort of forming the shape around the nucleus of the cell. During my postdoc, I also did some other studies with tau, but for the sake of time today, I'm going to focus on the work that we did with the peptide amyloid-beta. Amyloid-beta is toxic to neurons, and it starts as this transmembrane protein called amyloid precursor protein. It's cleaved by a series of other proteins, beta secretase and gamma secretase, to produce this soluble peptide. Now, this peptide can be a variety of different lengths. The one that we did all of our experiments with was A-beta 40, so 40 amino acids in length. 40 and 42 are the most commonly found in biology, And 40 is just a little easier to work with because it doesn't aggregate quite as fast. So this monomeric amyloid beta exists natively in healthy neurons. Presumably it has some native biological function, and that's no problem. The problem is when a lot of this A beta starts to accumulate, and it can form aggregates and eventually those large-scale amyloid plaques that we can detect in brains. Amyloid beta goes through a very specific, well-characterized aggregation pathway, where it starts as this intrinsically disordered monomer and then aggregates into these fibrillar oligomers, fibrillar meaning these beta sheet-rich structures that are fairly small in size. And these act as seeds for the development of mature fibrils, some more beta sheets, very stable. We can observe these fibrils by electron microscopy, and on a macro scale, this is what makes up those amyloid plaques. Another pathway is for it to aggregate into non-fibrillar oligomers, I'll call them NFOs, which are globular. They are not beta sheet rich. They're larger and more heterogeneous in size. And it is thought that these can perhaps convert to the fibrillar form. They can disaggregate back into monomer and then convert to fibrillar form. But what is fairly well agreed upon by most folks at this point is that these oligomeric species are the most toxic species when it comes to Alzheimer's disease, because in fact we have observed amyloid plaques in the brains of healthy individuals, and so we really do think it's the oligomers at this point. And in the literature, there's not always a clear distinction between which type of oligomer the authors are working with. They often say oligomer. So I'm going to be very clear in this study. We mean fibrillar oligomers made of beta sheets, or non-fibrillar oligomers that are more globular. So now to the involvement of the membrane in this process. The membrane plays an important role because it catalyzes fibrillation. This has been shown that monomeric A beta inserts into a cell membrane, and that insertion event nucleates fibril formation, first in the shape of oligomers and then in those macro scale fibrils. So this oligomer formation can also happen in solution, but it's a much slower process. And there's also evidence that these oligomers can interact directly with the membrane. And the end result is degradation of the membrane. And when I say degradation of the membrane, I mean macro scale disruption. So in this, there are many ways that this has been demonstrated, but this is one of my favorites because it's so striking. Where in this top row, we have A beta monomers interacting with different vesicles, and there's just 3 different representative images showing that. And in these cases, the vesicles look fairly intact. The fibrils, as you see something similar, where we add A beta fibrils to these vesicles, they still look fairly intact. But it's a different story for the oligomers, where they cause severe deformation to the membrane. We can see it in these blebs and chunks of membrane coming off of the vesicle. And dye leakage experiments have also shown that this compromises the ability of this membrane to act as a barrier. So not only is there this obvious deformation, but that critical function of the membrane being a semi-permeable barrier is also destroyed based on these A beta oligomers binding. So that gets us to the main question for this first study, is comparing intrinsically disordered monomeric A beta with the FOs and the NFOs. How do these structures interact with lipid membranes? What's different and what's similar about them? And once they do interact, how does it change the structure of the protein? And how does that change the structure and behavior of the membrane? The first step was to prepare these different amyloid beta constructs and confirm that we really had the starting materials we thought we did. And this is a fairly artificial preparation, but these types of structures have been previously isolated from brains, from AD brains, and this preparation was developed to mimic what was observed naturally in biology in diseased brains. So first we did electron microscopy, just to confirm that we could see these non-fibrillar oligomers. They're supposed to be bigger in size and more heterogeneous, which is what we saw. We did dot blot, where first the 6E10 antibody is just for the amyloid-beta sequence, so the antibody should bind all 3 cases, and it does. The OC antibody binds fibril structure, so we expected a more intense dot for the FOs. It's a little messy, not completely convincing. The A11 is an antibody for non-fibrillar oligomer, and that one was fairly specific and really confirmed we made the structure we intended. We also confirmed this by circular dichrosome spectroscopy to get an idea of the secondary structure of these different constructs. And the amyloid-beta monomers and the NFOs both had a signature peaks for being random coil, and the FOs had this very signature beta sheet peak. So with this, we felt fairly confident that we were starting with the constructs that we intended to. Next, we tested these constructs and measured their insertion into an anionic membrane using Langmuir Trough. We chose to use DMPG, which is an anionic lipid, and this is because previous studies have shown that A-beta prefers to interact with anionic lipids. And what we see is that all 3 protein constructs caused an increase in surface pressure, indicating that they're interacting. A-beta monomer had a lag time of about 1.5 hours. FO inserted right away, and so did NFO. But what was really interesting is that NFO then showed a decrease in surface pressure, indicating that some material was leaving the lipid membrane. So maybe it was destabilizing the membrane, which would make sense with that detergent-like hypothesis. We also did fluorescence microscopy. So looking at the interactions of A-beta monomers over time, we see these bright patches develop on the outside of our LC domains, and then eventually they become less distinct and overall more LE phase. In the FOs, we saw something sort of similar, where it starts with these bright patches on the outside of the LE domains, the LC domains, and then overall more liquid expanded phase was observed. And the NFOs looked pretty different, where we see these bright patches developing on the inside of the LC domains. So what do we make of these bright patches? They can be a couple of different things. The one that makes the most sense is, or one of the classical things we know can cause this, are lipids coming in and out of the plane of focus for the microscopy. So this would make sense, for example, if the membrane was invaginating, if we were losing material at the membrane, which tracks really well with what we observe for the NFO. So that's how we interpret these bright patches being formed for the NFO. That explanation doesn't make as much sense for the A-beta monomer and the FO, and we wonder if somehow the Texas red is being concentrated in local positions, but we weren't really sure entirely how to interpret that data. Next, we performed X-ray reflectivity, and this is what the pure DMPG looks like, where we see this peak for the electron-dense lipid heads. For the A-beta monomer and the FO, we observed this clear addition of electron density bound beneath the lipid head groups, and this additional layer of protein had a length of about 34 to 37 angstroms. Surprisingly, we did not see anything like that for the NFO. In fact, by this XR data, there is very little evidence that NFO was present at all. I'll also point out, just showing the table of our fit parameters for the XR. I realize this is a lot to look at, but I just want to point out the tail thickness and how it decreased with addition of A-beta monomers or FO. So this makes sense where we're seeing more liquid expanded phase on the microscopy. We would expect that liquid expanded phase to have shorter tail lengths, and that's exactly what we see with the XR data. Next, we perform GID, and this main peak that you see here is diffraction from the LC domains and the lipids. And overall, we don't see a change in the lattice spacing, but what we do see is a drastic decrease in the peak area, so indicating that overall we're developing fewer LC domains, and also a decrease in the coherence length. So the domains are also becoming smaller as we're adding protein. And we saw the biggest effect for reduced peak area with the NFOs. I'd like to draw your attention to this small peak here, which is highlighted in this inlay. So for FO and A-beta monomer, we observed a second diffraction peak. And calculating the d-spacing for that peak, we find that it's 4.7 angstroms, which is exactly what's known to be the spacing between beta strands and a beta sheet for amyloid fibrils. So we believe that we're detecting amyloid fibrils, or these small beta sheet-rich structures, at the membrane surface. So for FOs, that was the same structure we started with, likely just binding the membrane. But for monomeric A-beta, we're observing transition from a disordered into this beta sheet-rich structure. We wanted to study these using a slightly more biological membrane. The monolayer is a wonderful model system because we can control so much about it, but it is in the end still a monolayer. So we chose to do some experiments using small unilamellar vesicles composed of a 7:3 ratio of POPC, a zwitterionic lipid, and POPG, an anionic lipid. And with these vesicles, we did circular dichroism spectroscopy to get an idea of the secondary structure of these protein constructs. So first, in A-beta monomer, we see the monomer alone looks intrinsically disordered. It has a distinct change in secondary structure upon interacting with the SUVs. Maybe the start of a beta sheet peak here, but really some mixed structure. The FOs started in a beta sheet conformation and ended in a beta sheet conformation, so overall not much change. And the NFOs started disordered and stayed that way, but do notice that the data became much noisier after we added the vesicles, and we think this agrees with the idea that the NFOs may be breaking bits of the membrane off and making a more, not cloudy, but making a solution that's more difficult for light to pass through. So it's creating all this noise in the signal. So overall, combining our XR and GID data, we can deduce an approximate model for what these beta sheet-rich structures may look like at the membrane. From XR, we saw that these had a width of about 36 angstroms. We saw from the coherence length, they had a size of about 70 angstroms where they coherently scattered. The distance between beta strands is 4.7 angstroms. And we know that as these beta sheet-rich structures interact with the membrane, they're causing the membrane to be more disordered, converting it into an LE phase. So our overall conclusions from this study were, when we start with a pure DMPG membrane, we observe stable lipid packing, this condensed phase. When we added monomeric A-beta, it converted the membrane to an expanded phase and also assembled into a beta sheet-rich structure. When we added fibrillar oligomers, it seems like that structure bound to the membrane and didn't change very much, making those same changes. So overall, this disrupted the lipid packing and also caused thinning of the membrane, which is an important feature for the permeability of the membrane to different molecules. The non-fibrillar oligomers, our best interpretation is that they are binding the membrane and extracting lipids in a detergent-like fashion. So we see fewer LC domains, most likely as a function just that we have fewer lipids there at the surface and that we're losing surface pressure. I didn't show this data for the sake of time, but I'll also point out that we did studies with those 3 protein constructs adsorbing to a pure air-water interface, no lipids present. And we observed the same thing in all 3 cases, where we had indication of a beta sheet-rich structure with a width of 19 angstroms, which is distinct from the width we observed on nucleated when lipids were present. So this indicates that the template for fibril formation makes a difference in the structure of those fibrils. So lipids will template one fibril structure, and the air-water interface will template a different fibril structure. We also observed a second diffraction peak, indicating that there was this 40 angstrom diffracting unit that repeated in space. So that was one of the applications of these liquid surface X-ray scattering methods. The next I'll talk about is this protein lipoxygenase, which is a peripheral membrane protein that binds and oxidizes lipids. First, I'd like to thank the folks who are working on this project. Yarrick has this great skill for bringing together different people with different skill sets to answer a scientific question, and that's exactly what's happened in this situation. Dr. Marcia Newcomer, she's a PI at Louisiana State University, and lipoxygenases have been a huge part of her career. She's sort of the lipoxygenase queen. So she's been initiating a lot of the ideas for these experiments, as well as a scientist in her lab, Dr. Nathan Gilbert, who's been really key in designing the experiments, also preparing the proteins and helping us collect the data there at the synchrotron. Of course, this work again was done at NSF ChemMatCARS. Way is amazing, helping keep the beamline running in great shape all the time, allowing us to collect our data. So lipoxygenases oxidize polyunsaturated fatty acids, or PUFAs. And when these PUFAs are oxidized, it can cause a lot of different effects in the cell. Some of our favorite anti-inflammatory drugs inhibit these protein cyclooxygenases and are involved in inflammation response. In a separate pathway, we have the lipoxygenases, the ones I'll talk about today, which there's a family of several of those that exist natively in humans, and inhibitors for certain LOX proteins are drugs that are on the market right now. So I'm going to talk specifically about one lipoxygenase called 15-LOX2 that has been linked to the development of atherosclerotic plaques. The way this works is that 15-LOX2 oxidizes lipids that are transported by LDLs, and then macrophages ingest those LDLs, and it transforms the macrophages into foam cells, and the accumulation of foam cells leads to development of these plaques in the blood vessels. So the idea is that if we can develop an inhibitor for 15-LOX2, this could be a viable therapy for mitigating cardiovascular disease. This is the crystal structure of 15-LOX2. It was solved in aqueous conditions, but there's a fair amount of evidence that this protein adopts a different structure as it binds the membrane. But we'll start with what we know from the aqueous conditions, where here's 15-LOX2, and it has these two key hydrophobic regions. The most important is this hydrophobic loop here, and the second is maybe one or two amino acids that are hydrophobic on the end of this helix. This hydrophobic loop is also thought to be stabilized by calcium ions that bind the base of it. And indeed, truncation of this hydrophobic loop when it's just cut off, or when calcium is absent, both of those things decrease enzyme activity. So we know that they somehow help the enzyme work better. The orientations of proteins in membrane server is able to predict how a protein might bind the membrane. And it basically uses the structure of the protein and looks at the surface exposed amino acids and calculates like a partition coefficient for the whole protein based on the identity of each amino acid. So the OPM server predicts that 15-LOX2 would bind the membrane with this hydrophobic loop buried in the membrane, and then those one or two hydrophobic amino acids. And so far, this is the best guess folks have for what it might look like. But as I said, there is other evidence that it might adopt a different conformation while binding the membrane. Here's that same 15-LOX2 protein, but in this image, we've highlighted a lipid bound into the active site of the protein. When that lipid is bound there, the substrate is particularly arachidonic acid with these multiple points of unsaturation in the lipid tail. And this is converted to a peroxide-modified arachidonic acid. That's what 15-LOX2 does. So it remains this question: how does the lipid up here, this arachidonic acid it needs to modify, how does the enzyme get the substrate into the active site for modification? And then what does it do with that lipid? Does it put it back? How does this process work? So far, there's not a lot of evidence for, any large swinging hinge domains. So we have this question of how does the enzyme access its substrate? Oh, and just to add, once these enzymes are modified by, once these lipids are modified by peroxide, the thought is that this hydrophilic peroxide group wants to associate with the head group, so it makes the overall surface area of the lipid expand, and this floats to the surface, which will be important later when we're interpreting our X-ray reflectivity data. So this brings me to our question, which was how does 15-LOX2 bind membranes? And then it binds and it adds this peroxide group. How does that lipid peroxidation impact the membrane structure? We designed our experiments using two different lipids. The first is DSPC, so you'll notice there are no unsaturated tails here, no substrate for the enzyme. The second is SAPC, where we have this one arachidonic tail that is the substrate for the enzyme. We used three different variants of the protein. One expressed very carefully to exclude calcium, and there is 0.5 millimolar EDTA in here, so no calcium could bind. The second is where we add in a small amount of calcium pre-mixed with the protein. I think it was 2 or 5 millimolar, so larger than the concentration of EDTA. And then the third is with a loop mutant, where this hydrophobic loop is truncated from the structure. So by using these different combinations, we're hoping to learn how membrane composition impacts protein binding, how the loop mutant impacts protein binding, and also the role of calcium. So this is Langmuir Trough data testing the insertion of wild-type 15-LOX2 into different membrane conditions. When we test insertion into a pure membrane made of DSPC, we see this increase in surface pressure over time, perhaps indicating LOX binding the membrane. When we test with a mixture of 8:2 DSPC to SAPC, we see this decrease in surface pressure over time, which we haven't fully understood yet. It could be attributed to a couple different things. One thing is perhaps that LOX is adding this peroxide group, which is causing the membrane to expand, and in our constant area system, there may not be room for that membrane expansion. So in fact, it may be causing sort of blebs and invagination in the membrane, which would look like a decrease in surface pressure. It's also possible the protein is extracting lipids from the membrane, although we don't think that's the case at this point. And when we worked with the pure SAPC membrane, we also see this decrease in surface pressure over time. Now these experiments need to be repeated to all start at the same surface pressure. We realize this could be a function of net surface pressure and not which membrane composition, but so far we're pretty excited about this data. We also used a constant membrane of 8:2 DSPC to SAPC and added different LOX proteins. We found that wild-type LOX causes this decrease in surface pressure. This is the same data as you see right here. And then when we have wild-type LOX with calcium, again, this decrease in surface pressure. And when we use the loop mutant, we do not see that decrease in surface pressure, and instead we see a slight increase. So to clarify, this wild type LOX is in the absence of calcium. All of these experiments were performed in the absence of calcium, unless it's explicitly stated that calcium is present. Here's the results of the reflectivity, where in black is the pure DSPC membrane, and in red is after addition of wild-type LOX. So we see maybe a slight increase in electron density of the tail, but not very much signal overall. With the pure SAPC membrane, we see this increase in density and shift. And with a combination of 8:2 DSPC to SAPC, we see this large increase in electron density in the tail region. So let's think about these changes that we're seeing in relation to the predicted binding of 15-LOX2. So first we have this small increase in electron density. Considering our membrane is pure SAPC, and one tail on our SAPC can be modified, this increase in electron density works out to about what you would expect for the addition of 2 oxygens for every other tail on these lipids. So we think this might be due to this lipid peroxidation. This also makes sense from this shift in electron density over where the peroxide group might be floating to interact with the head group. We don't really see evidence of this stably bound protein in the pure SAPC membrane. Although, when we take the structure of this protein and we calculate the expected electron density, we get something that matches about what we see for the lipid head groups. So it's a little tough to discern in this case. Highlighting again the 8:2 DSPC SAPC. In this case, for only 1 out of 5 lipids is an SAPC, which means 1 out of 10 tails is a substrate for the enzyme. There is no way that the addition of these oxygens can cause this increase in electron density. So we're fairly convinced at this point that this increase has to be due to protein binding. And again, we don't think we can distinguish the protein from the head groups, and we don't see any evidence of the protein binding beneath the head groups. So this suggests that this model generated by OPM might not be telling the whole story. We think the protein might be binding, submerging into the membrane, occupying a total length of about 30 angstroms, compared to when you measure this protein along this axis, it has a length of closer to 40 or 50. So the protein might be extending along its long axis, compressing here and embedding into the membrane. Here's the impact of the calcium and loop mutant, which was actually very small. So all of these experiments were done in 8:2 DSPC to SAPC, and we can't really tell much of a difference at all with the loop mutant compared to wild type. And when calcium was present, we see just a small additional increase, which may be due even to the presence of calcium as a strong electron scatterer. So we're not entirely sure, but overall, not much difference from changing these factors. Again, here is our complete table, which is a lot to look at. But what I want to point out is that when we used a membrane that had DSPC in it, we see a lengthening of these lipid tails by an angstrom or more, which is quite significant. So this is telling us that the 15-LOX2 is definitely interacting with the DSPC, causing it to be the tails a little more extended. And we thought it was very interesting when we confirmed that observation with GID. So here in black is the pure DSPC membrane, and in orange is after addition of wild-type LOX. And we see this shift in the peak, which results in a compaction. So we can use this to calculate the area per lipid, and we see that overall we have this decrease of about 0.5 angstroms per lipid, which is fairly significant. You'll also notice that there is lower area, which indicates fewer lipids in the LC phase overall. So we're observing that LOX binding causes compaction of the lattice spacing and also fewer lipids in the LC phase. The pure SAPC membrane didn't diffract on its own, which was not entirely surprising. So we couldn't really tell much from this data. And then the mixture of 80/20 DSPC SAPC looked fairly similar to the pure DSPC, where we observed compaction of the crystalline lattice and also reduction in peak area. This got a little messy when I tried to overlay these four, so instead I have them separate, but you can compare the area per lipid when we added calcium and loop mutant and the peak area. And in all cases, we see the same theme of compaction of the lattice and a reduction in peak area, indicating that lipids are being converted from the LC to the LE phase. And while there were subtle changes among the calcium and loop mutant, overall it was really the same trends as the pure wild type LOX. So overall, we find that 15-LOX2 binds membranes and interacts with lipids, regardless of the presence of this hydrophobic loop or the presence of calcium. We find that the surface pressure decrease is not observed when the hydrophobic loop is absent or when there's no arachidonyl substrate. So we think that decrease in surface pressure as a result of protein binding might be significant. We found that 15-LOX2 interacts with lipid tails, causing them to pack together more tightly, and it also causes lipids to convert from LC to LE phase. We have this preliminary evidence that there may be a major conformational change occurring when 15-LOX2 binds the membrane. Where the aqueous structure measures 40 to 50 angstroms in width, we're seeing something that's closer to 30 angstroms. So in the future, we need to repeat these Langmuir Trough studies, and we'd also like to add fluorescence microscopy to get a better idea of that domain separation using another method. We're going to perform mass spec to verify the presence of that peroxide modified lipid product. We would like to perform X-ray scattering with an inactive version of the protein where the native protein has iron bound at the active site. We can use a form that has manganese bound at the active site, and it allows the protein to bind the membrane but not to do any catalysis. So this would really help us pick apart those changes in electron density that we're seeing and address, are they caused by protein binding or by lipid peroxidation? And another control we'd like to do is to use a chemically oxidized lipid, so I have no proteins in the system at all, and chemically oxidized, again, to help pick apart those two factors. So again, I'd like to acknowledge the folks who were part of this project. For the amyloidogenic peptides project, that was work I did in my postdoc with Dr. Eva Chi. For the 15-LOX2 project that's ongoing, that's work that I'm doing with Dr. Yaroslav Majewski and the newcomer lab at LSU, including Dr. Nathan Gilbert. And then finally, I'd like to thank the hardworking undergraduates and master's students in my lab at UCCS. It's a special circumstance to be doing research where no one is paid to do research full-time. Everybody here has other obligations for teaching. A lot of my students work on the outside or their parents, so they're some of the most hardworking people I've ever met, and they're very committed to getting the work done. I hired Cosme on this semester, and I'm training him on the Langmuir Trough right now, and he's going to pick up this lipoxygenase project and do all these future experiments. And then finally, for the live version of this talk, the seminar was occurring during a time when I was scheduled to teach. So the unsung hero of this talk is Dr. Wendy Hagren, who covered my teaching for that morning. So thank you all for listening. I did not include my email address on this slide, and I should have. It's cvanderz, cvanderz @uccs.edu. So please feel free to reach out to me if you have any questions or if you'd like to have a discussion about the talk. Thank you for your time.